All right, it looks like we are ready to begin. So let's get started. So hello again, and welcome to Biocytogen's webinar series. It's great to have you all with us online today. So in today's session, we will be focusing on autoimmune indications, specifically atopic dermatitis and asthma, and we will be discussing how to choose the right in vivo models to support therapeutic development for those indications. So before we dive in, let me briefly introduce myself and our team here. So my name is Yuan Tian, and I am the marketing manager at Biocytogen Boston Corporation. I will be serving as your moderator today. And I'm also joined by two amazing colleagues from Biocytogen who will be participating in the Q&A session later on. So with us today are Dr. Yujie Liu, Director of the Biomass Preclinical Services Department, and Dr. Lan Xiaoxiong, who is a business development manager at Biocytogen. So thank you both for being here today. We're really excited to hear your perspectives. And next, it's my pleasure to introduce Dr. Zhen Chen, who will be our speaker for today's webinar. So Dr. Chen currently serves as the head of the autoimmune pharmacology department at Biocytogen. He received his PhD in biology from Nanjing University, where his research focused on neurodevelopment and regeneration, building a strong foundation in genetics and humanized animal disease models. And since joining Biocytogen, Dr. Chen has taken on several key leadership roles, including senior scientist in model development and senior study director. So today he leads the autoimmune pharmacology division, where he focuses on developing innovative humanized disease models and conducting high quality preclinical studies. And over the years, Dr. Chen has managed over 100 preclinical projects and has supported multiple pharmaceutical partners in successful IND and NDA filings in both China and the United States. So we're very excited to have him with us today to share his insights. All right, with that, in today's webinar, Dr. Chen will be talking about Th2-driven inflammatory diseases like atopic dermatitis and asthma, and we will introduce Biocytogen's target-humanized mouse models for these indications, highlight several clinically relevant targets, and share practical insights and key studies on the selection and application of these models to support drug development. All right, so before we begin, just a quick reminder, the Q&A box is open, so feel free to send in your questions at any time during the session. We will save time at the end to address as many questions as possible. All right, without further delay, I'll hand things over to Dr. Chen.
Thank you for your introduction, Yuan. Hello, everyone. My name is Jen from Biocytogen. It's a great honor to share how our humanized mice model are accelerating drug discovery for AD and asthma. We are at a turning point. In the 20th century, we found external infections, but in the 21st century, the challenge is chronic systemic inflammation. As average lifespan increased from 60 to over 80 years, the unmet clinical needs for autoimmune disease are growing fast. We must move from generic killing to precision targeting, and from treating diseases to maintain long-term health. The shift requires us to understand the true nature of autoimmunity. Is the disruption of pathogenic cytokine networks and the loss of immune tolerance. Whether diseases are organ-specific or systemic, the underlying immune imbalance is the same. Atopic dermatitis, AD, and asthma are localized eruptions of this systemic Th2 dysfunction. And because of this change, the market is exploding. By 2033, the global autoimmune market is expected to reach $137 billion. Looking at 2024 license deals, complex biologics like ADC and bispecifics now account for 44% of the total deal count, and the upfront payment account for 66%. These complex drugs offer better results, but they also create a trap for R&D because they target human-specific proteins. They show almost no activity in standard wild-type mice. So to de-risk these drugs before they enter clinical trials, we urgently need high-fidelity humanized mouse models to bridge the transcriptional gap. And we have discussed the microtrends and the need to de-risk development. Now, how do we apply this to your specific pipelines? My presentation will follow this three-part outline. First, target overview. We will provide a comprehensive look at humanized targets for Th2-mediated autoimmune diseases. This is the foundation of our research. And second, asthma insight. I will share in vivo efficacy data for emerging targets in our asthma models covering both MABs and bispecifics. And third, the AD evolution. We will use case studies to explore evolution strategies for MABs, bispecifics, and even the latest ADC drugs in AD models. Now, let's begin with part 1, an overview of humanized targets for Th2-mediated autoimmune disease. Okay, let's look at the mechanism first. The biologic core of the asthma and AD is the cause organ Th2 pathway. As shown in this slide, signalings follow from upstream laminin-like TSLP and IL-13/3 to downstream effectors like IL-4, IL-13. The big problem is that these cytokines and their receptors are highly specific to each species between human and mice. This difference causes a major translational gap. Based on the data, AD and asthma remain critical high incidence diseases, affecting about 600 million and 400 million people. Where benchmark drugs like dupilumab has proven the value of IL-4R receptor target. And this show almost, this show almost zero activities in standard wild-type mice due to binding site differences. Without humanized model, your efficacy and the PK/PD data may be invalid for clinical translation. And to give you a clear sense of the current market hit, let's look at this 2026 monoclonal antibody R&D track. This isn't just a list. It represents a massive global commitment to Th2-mediated pathway. This field is highly validated with multiple blockbusters. For example, IL-4R, led by dupilumab, not only dominant AD and asthma, but also secured approval for COPD in 2024, showing real staying power in the market. And other targets like IgE, IL-5, IL-5R, TSLP are already launched and saving millions. And what's more exciting is that the second wave in the pipeline. They have several high potential targets in phase 3. such as IL-13, and OX40, OX40L, which are very close to major data readouts. And look at the long list below, like IL-25, tier 1A, it also shows the industrious is going or in the autoimmune diseases. And the Th2 market is proving and growing, but the race is on. Since these top drugs are built strictly for humans, have high quality human nice models, mouse models isn't just an extra. It's our so is your ticket to play and avoid the translational gap. And to solve the translational gap, Biocytogen has built the complete humanized mice library. Think of it this way: if our mice are the seeds, then our disease platforms is the soil. You need both to see the fruit, which is your efficacy data. And our platform covers skin, lung, gut, and other systems with ready-to-use the protocol for things like asthma and skin inflammations disease. The K is here. I think the K here is the readiness. Every models in green comes with proven PD data. This all-in-one solution cuts your risk by giving you results you can trust. And in 2026, R&D has shifted towards bispecifics and combination therapy. To stay ahead, you need more than standard tools. We provide an off-on-shared catalog designed for this area beyond the IL-4 R double humanized mice. We also offer 5 and even 6 target humanized mice models covering pathways like IL-4, IL-13, OX40, all at once. And these are ready to ship inventory, not just the idea on the paper. In the speed first market, these complete models allow you to start testing multiple pathways validation and reach milestones faster than the competition. And now let's move to section 2, asthma insight. We will use specific in vivo case studies demonstrate how humanized mice models build a solid proof and reduce your clinical translational risk. And the Th2 axis is at the heart of the asthma. As shown here, allergens active with the DC cells, leading to the Th2 cells differentiation and cytokines productions. IL-4, IL-13 drive the IgE productions and eosinophil infiltration and mucus production. IL-4R, the shared receptor, is the best treatment. And why we focus on this target? Dupilumab is proving a massive success with 2025 revenue expected to reach $18 billion. Is amazing. And however, it has nearly zero activities in standard wild-type mice. And so to de-risk your projects, we developed our flagship IL-4R double humanized mice model. Our precise replacement strategy ensures the drug binds to both humanized ligand and the receptor, which is critical for testing these strong antibodies. And a reliable model needs a clean background. And as you can see, we confirm specific human IL-4 and IL-4 receptor expression in mRNA and protein levels with zero mouse counterparts. And flow data further show that the development of TBNK cells is identical to the wild-type mice. This ensures your drug is tested in health and a natural immune environment. But presence does not always means performance. To prove the model actually works, we conducted functional validation. In our ex vivo assay, we demonstrate that human IL-4 successfully triggered the B cell IgE production in these mice, and more importantly, this biological process can be blocked by dupilumab in a dose-dependent manner. This confirms that the humanized targets are not just expressed, they are biologically active and fully responsive to human-specific antibody drugs. This is the gold standard proof you need before moving into in vivo efficacy trials. And in our OVA-induced asthma model, dupilumab treatment leads to significant drop in both eosinophils and serine IgE levels, including the total IgE and OVA-specific IgE. And histopathology data also shows in the modeling group, there has a severe infiltration and mucus production. And the treatment group shows improvement. We have discussed the monoclonal antibodies, but the market is moving fast to the bispecific era. Why? Because blocking a single target often hits a power limit. To break through, the industry is now targeting multiple pathways at once to lower clinical risk. And look at our updated 2026 industrial tracker. We can find that there has a breakthrough like the Acacia's AK139. That's a IL-4R and ST2 bispecific antibody was approved for phase 2 trials in amazing seven indications this February, and is now the world's leading candidate for this combination. And but as we know, the road to success is paved with lessons. For instance, J&J's decision to stop the NM26 in late 25 remind us that the bispecific R&D is a high-risk game. And this table shows that everyone is going all in the multiple targets, from Sanofi to Pfizer, but I think that without the right tools, you are flying blind. Confirming that the extra powder in multiple targets model is no longer a choice. Is your insurance policy against clinical failure. Now let's see how our multiple targets humanized mice provide that exact proof. For candidate targeting upstream alarmins, our IL-4, IL-13/3, TSLP, TSLP-R, triple humanized mice is a good standard. We tested the combination of itepekimab and tezepelumab. Group 5 showed that near perfect anti-inflammatory cells and eosinophils. They also show total IgE levels and some downstream cytokines like IL-4, IL-5, IL-13 track were almost suppressed by the drug treatment. And furthermore, histopathology data also show that the airways return to even normal state in this after the treatment. And I want to take a moment to highlight our practical experience on this model. We have already evaluated multiple bispecific programs use this specific model, and the efficacy data is highly encouraging. Where this bispecific data remain confidential to protect our partners' interests, and but it confirms that our multiple targeted models are fully validated for complex new drugs. Yeah. And to wrap up the asthma section, choosing the right soil for your seeds is critical de-risking step. Here is our strategy advice for your model choice. For the antigen-induced asthma models, these are the gold standard for chronic studies, since they involve an active immunosensitization phase. They are essential for fighting airway remodeling and long-term inflammatory memory. Choose these if your drugs aimed for disease modification, and for the cytokine-induced asthma, think this as your first track for target-specific validation. By skipping the long sensitization process, you get a clean and high contrast signal. This is a game changer for complex bispecific, saving you 2 to 3 weeks of turnaround time. And then in 2026, we increasingly to recommend a hybrid approach, using cytokine-induced model for rapid MOA confirmation, and followed by antigen-induced asthma for tissue proofs. And this dual package is exactly what regulatory agencies look for to ensure clinical success. And now that we have covered the lungs, and how does this same immune fires show up on the skin? Let's move to the skin. AD is often called skin asthma because it shares the same Th2 driving the signaling. In section 3, we will use case study to explore evolution strategy for MABs, bispecifics, and the latest ADC candidates in our AD models. And let's start it with dupilumab. In our OXA-induced AD model, we use a well-established protocol starting with sensitization on day 0 and challenger from day 7. The data is very clearly, and we observe that a significant drop in the ear thickness and the serum IgE levels after treatment by dupilumab. And please look at the HE slices. You can see that the thick epidermal thickness proved, and this visible efficacy is the most direct evidence we provide to de-risk your drug before clinical trial. And as a professional partner, we provide deep molecular insights for your IND submission. This slide showed time-dependent IL-4 protein expression, and the dupilumab treatment significantly lowered the IL-4 levels in both ear and skin tissues, particularly during the late phase from day 16 to 26. And combined with dynamic RNA-seq analysis of Th2 genes, this time course data is extremely useful for helping you future study using frequency. Yeah. And finally, we use flow to profile the immune cells population in the ear. The results show that treatment significantly reduced the not only the eosinophils, but also T cells and Th cells, and especially the Th2 cells subpopulation. This deep dive analysis explains the biological logic behind your drug's success. And when we discuss ADCs, we usually think of oncology. However, in 2026, ADCs are becoming a game changer in the autoimmune field by directly targeting and clearing bad immune cells. They offer the potential for long-term healing. And let's look at how our platform supports de-risking these cutting-edge drugs. Now let's talk about ADCs. In this study, we tested 2 ADCs drugs in our humanized mice. Please look at the graph A, ear thickness, where the modeling groups went up, and the ADC 2 treatment groups stay almost flat for the whole 26 days. And in the graph B, the Ig levels for ADC2 treatment was also very low. Move to graph C. ADC2 generally greatly reduce the epidermal thickness and have the lowest TOR scores. And you can see that the ADC2 clearly did better than the benchmark drug, dupilumab, the group 5. And with a strong p-value, ADC2 proves it has huge power to stop these diseases. But however, we must also look at safety. That also shows the body weight data for the same study. And look at graph A, body weight. To be honest, the ADC2 treated group, the green line show a sharp drop in the weight earlier. It fell below the 19% lines and stay lower. We view this weight loss as an early red flag for safety. Where the drug is very strong, this body weight drop suggests a smaller safety range. And this is the value of Biocytogen, de-risking. By finding these signs earlier in our humanized mice, we can help you to make smart choice, like fixing the molecules or changing the dose, long before you start expensive safety tests. And as Th2 research moved into more complex area, one test is often not enough to show a drug's full power. For bispecific drugs, we use for main readouts, ear thickness, serum IgE levels, histopathology, and itch reliefs. This helps us to prove that the drugs work together and are useful for patients. And to solve this, we developed 5 target-humanized mice. In our evolution of OX40L and IL4R pathway, the data prove that humanized mice is essential to verify the multiple targets synergy in ear thickness and IgE levels. And furthermore, the pathological score also shows the synergy effect. And we have already evaluated multiple bispecific program use this specific AD model, and the efficacy data of bispecific we have seen is highly encouraging. And however, this data remain confidential to protect our partners' interest. Okay, and in another study evaluating different targets combination, the IL-13-1R and IL-4R combination, we focused on the itch relief as the core functional readout. By using our 5 target humanized mice, the IL-13-1 and IL-4 target mice, we can clearly measure the scratching behavior. We recorded scratching behavior in these specific models. The results show that the combination of IL-4R and IL-13-1R reduce the ear thickness and scratching frequency over 60 minutes. And this behavior and pathology and molecular proof supported by a decrease in IgE levels and IL-4, IL-13-1 protein levels, and also in the histopathology scores. This can help build trust for clinical translation. Okay, we are now coming to the end of today's session, and the core thing is clear: bridge the gap between preclinical research and drug delivery. In 2026, with the rise of complex biologics, targeting specificity means that you can no longer rely on standard Vitamize model. Our biomass platform provides a total solution by combining humanized mice like IL-4, IL-4R, and other 5 or 6 target humanized mice, and these mice with validated disease model. And this allows you to identify the traps earlier and truly de-risk your project. And this slide summarizes our current capability metrics. And please look at these check marks. Each one represents not just a success case, but a ready-to-use off-on-shared resource available to you from TSLP, IL-13, IL-4R, TSLP, OX40L, IL-13, and IL-31. We have established benchmark data for MABs, bispecifics, ADC, and biologics. We provide much more than mice. We delivered high quality data and MOA insights and the data package for your IND. And fighting new drugs is a search for trust and results. Biocytogen wants to be your partner. We use reliable models and top quality data to help you go to the last mile from lab to the clinical. And thank you for your time. That's all I would like to share today, and I'm happy to take your questions. Yeah.
Thanks, Dr. Chen, for that great talk. That was really insightful. I saw that we already have many questions in the Q&A box, so I'll take it from here as we move into the Q&A session. And to our audience, if you haven't submitted your questions yet, feel free to do it now. And our panelists, Dr. Liu and Dr. Xiong, are also here to weigh in. All right, let's start with the first question here. So how long would it take for you to create a double knocking mouse from your existing single knocking mouse?
I can answer these questions. For creating the double knocking using our single knocking mouse, generally 5 months. Yeah, if you have specific models you want to cross, feel free to let us know, and we can provide a detailed timeline for you.
Perfect. Thanks, Wenqiao. And going to the next question. Can your asthma models monitor downstream biomarkers?
Yes, this question, I can take this questions. Yeah. Absolutely, yes, we can do this. And as shared in this presentation, I have shared some case studies like for the biomarkers like IgE, IL-4, IL-5, IL-13, and other biomarkers. And the second, this is the molecular biomarkers, and the second is the secular biomarkers. We can use the flow to test the eosinophils or neutrophils and other immune cells to evaluate this efficacy. And the third, we can detect the tissue from by histopathology methods to evaluate the efficacy. So combined with molecular, cell, and tissue biomarkers, we can detect that and provide service for you. Yeah.
Great. Thanks, Dr. Chen. And here's another question. Do you have any translational efficacy data from humanized mouse models to clinical applications for a given disease modality that you guys working on?
I can try to take this question. Yes, we do. As today Jen showed here, there is a lot of data that we have already generated, and we also generated a lot of data, which is our clients' data, and we help their studies for their R&D application. And just now, Jen showed that there is a lot of big deals last year, and some of the deals was also based on our data.
Great. Thanks, Dr. Liu. And going to the next question here, which asthma model do you typically recommend for chronic studies versus mechanistic testing?
Yeah, okay. This is a very strategic questions. And from my point, I think that it depends on your targets. And actually, we may recommend a hybrid strategy to use two models, both asthma and antigen-induced and cytokine-induced. For these two models, if you use studies focused on the chronic studies, or we may suggest to use the antigen-induced models. These are the gold standard for tracking long-term biomarkers. And but if you would like to test the mechanism and validation, the MOA, we may suggest to use the cytokine-induced asthmas. And because this is, this is your like a fast track for MOA confirmation, and it can provide a clean and high contrast signals for target-specific biomarkers. And once more, that can save you time, even 2 or 3 weeks. Yeah.
Awesome. Thanks, Dr. Chen. And going to the next question, which I think I can answer pretty quickly. Will you share the PPT at the end? So we will be preparing like the PPT shareable version after the webinar. So if you're registered for this webinar and if you're attendee, you will be receiving like a follow-up email with the shareable PPT after this. All right. So going to the next question here. Consider immunogenicity of target effects and toxicity, which approach like ADC, bispecific antibody, or biologics would be more safer and effective in your opinion?
Yeah, it's really a challenging question. For the safety effect and off-target effect, it really depends on what kinds of target you are going to study out, what you are going to target. So some targets is really clean and they are really easy and safe to be used in the clinic. And another thing is about the mechanism. Some bispecific antibodies to reduce the toxicity, but some of them may help to improve the immunology. So it really depends on what kinds of target you are using and what kinds of mechanism are depending on based your test article. But for example, ADC is a really good test article, and we can see that the efficacy of the ADC is really good, even better than the positive control. But sometimes the payload is toxic to the patient. But nowadays, we also know some payload that is not toxic at all, and is really a good kind of a good of biologics for the future evaluation. Yeah.
Thanks for the detailed answer, Jie. That was really helpful.
Thank you.
All right. Here is another question. Where did you perform this AD experiment, Boston or Beijing?
I think I can answer this question. So for AD models, we can do it either at Boston site or Beijing site. And so either location, we are able to do it. The data are shown here, I think is in our Beijing site, but we also have data from our Boston site if that's of interest.
Great, thanks, Nanqiao. Okay, here's another question. With the rise of ADCs in autoimmune diseases, what advantages does your model offer for drug evaluation?
I can try. So for the autoimmune disease models, generally there are 2 kinds of model. One is target-humanized mouse model, which is introduced today by Jen, and the other one is the immune reconstitution model that is mainly based on HSC reconstitution model or PBMC reconstitution model. We prefer immune competent mice because it can better monitor the better try to eliminate what happens in the human, and it has the most competent immune cells in the mice. So that's why we prefer to use these kinds of models for antibody or ADC evaluation. Yeah.
Great.
And meanwhile,
Thanks,
you can also see if the ADCs have some toxicity. This kinds of mouse models is much better to show to you because they have the most competent immune cells.
Great.
Yeah.
Thanks, Yujie. That is a very detailed answer. Let me finish this one. And going to the next question. How do you support clients in preparing for IND submissions?
I can try to answer these questions. I think from our experience, we will separate it into two aspects depending on the target, either the mature targets or more novel target. And for these IND submission, as you know, we follow the chain of evidence. And for the mature targets, such as an IL-4 receptor and a TSLP in asthma, we believe that the speed and benchmark are two important criteria. And as you can see in earlier in Jen's presentation, we use DEPU for DUPI for the benchmark molecules, so you are able to do head-to-head comparison. And that's what we would like to recommend. And we're also able to start the study without delay. I think for mature targets, speed is very important. And with us, we can start your study as soon as possible. And for some like novel and first-in-class molecules, we think not just speed. Another thing we need to add is the mechanism, and we are able to provide a very comprehensive MOA analysis and characterization in our study, in our package. And I think lots of clients will appreciate us identifying like potential safety red flag using humanized mouse. And some of those red flags like lost weight or some toxicity will be easy to more relatively easier to be detected using our humanized mouse. And I think that's the two aspects for the mature and the novel target. Another thing I want to add is that we're able to provide things regulatory ready. As Jen mentioned, we supported lots of successful IND approval using our data and models. And so those are we're able to provide a comprehensive data file while maintaining the good quality and the speed. And I think hopefully that answers the question about the IND.
Yeah, that's really helpful. Thanks, Lan Qiao. And I saw that there are several questions about the FcRn, so I'll read one of them just to give us an opportunity to discuss it a little bit. So this question here, have your several humanized mouse model crossed with human FcRn mice for PK and PD study together?
Yep. I think I can add for the FcRn, you're right, our FcRn mouse is very popular. And I believe with those FcRn, we do have the mouse available. And then, yeah, for FcRn.
Yes, we have
Great.
crossed FcRn and IL-4 receptor humanized mice, and this mice may will be available in 2026. Yeah, and prove the data.
Great. Thanks, Lan Qiao. Thanks, Jen. Let me finish this one. And here is another question. What are the key differences between OXA-induced versus KLH-induced AD models? Could you explain what key considerations should be when we choose between the OXA versus KLH-induced AD models?
Well, I can try. OXA-induced model is strongly suitable for atopic dermatitis research with skin barrier disruption, Th2 bias, and AD-like lesions. KLH-induced model is more like adaptive immunity or DTH evaluation, not a true AD model. So if you want to evaluate AD-related pathway, so OXA induced model is better than KLH model.
Great. Thanks, Yujie. And here's another question. Behavior data can be variable. Can scratching data, like what you presented, support an IND submission?
Yes, maybe I can take this question. Yeah, it's a very you're right. This the scratching behavior shows us some variation in the group, but actually is a very is a very clinical result related to patients. So we may think that we provide the different level result like ear thickness, IG levels, cytokine levels, and his pathology. And this scratching behavior can be a more clinical related result to support this data package for you. Yeah.
Great. Thanks, Dr. Chen. And I also saw several questions talking about the mouse background. I'll read one of them to discuss it a little bit. So, so here's a question. Are most of your knocking mice from B6 background? Do you offer knocking from biopsy as well?
I can quickly answer. The answer is yes. We have the models in biopsy as well, and most of our models from Black Cyst, you're right, but we also have for same version like biopsy, for example, IL-4 receptor double humanized mouse. We have those available in biopsy and many other strains.
Awesome. Thanks, Lanqiao. Okay, going to the next question. How do you choose dosing for bispecific or combination therapies in efficacy studies?
Yeah, I think I can take this question. It's a very important question before we take a study design. And actually, our clients may ask question, and they may have this choose for bispecific. They may choose the molar equivalent dose compared to the monoclonal antibodies. And for the combination therapy, that has two methods. One is the sub therapeutic dosage, like the low dose plus low dose to compare the monotherapy to get a good efficacy of synergy. And another is this combination is very strong and has very good efficacy, and they can compare to the high dose of the mono therapies to get more data. But actually, this is I think this is some rules for the dosage choose. But actually, we need to considering more about your drugs. And like if you are a bispecific antibodies, you may need to consider the PK and the half-lives compared to the mono antibodies. Usually, bispecific antibody has less has lower half lives, and also need to considering about like FC effectors and other effectors about that. So this is a case by case choice for this. Yeah.
Awesome. Thanks, Jen. I think we're running a bit short on time, so I'll take two last questions. So one question here is, for your knocking mice with 4 or 5 human genes, are they all homozygous without any mice genes?
I can chime Jimin. I think for the knocking mouse, yeah, they are homozygous. Like if you're asking the humanized IL-4 receptor, with a humanized, like for example, IL-13 receptor, and those are homozygous. Yeah, if you want, I think for some transgenic mouse, depending on the knocking strategy, they may be different, but you can, for most triple knock-in, 4 gene knock-in, 5 gene knock-in, they are homozygous. If you prefer to test with heterozygous, also feel free to let us know. We are happy to be like accommodate.
Great. Thanks, Lanqiao. So the last question for today, can you explain how the precise replacement strategy works in the IL-4RA double humanized mice?
Okay, I can take this question. As to the IL-4, the exon 1 to 4 of the mouse IL-4 gene that encodes full length coding sequence was replaced by the human IL-4 gene in the humanized mice, and the exon 4 to 7 of the mouse IL-4RA mouse gene was encoded. The extracellular region coding sequence was replaced by the human exon 4 to 7 in the IL-4RA double humanized mice. And after, so the promoters of the mice are all the mice. So the expression pattern of the protein is the same after the humanization. And then we do the validation based on the gene level and the protein level. So we detect if the IL-4 express the same level in the wild-type mice and in the humanized mice, and the result is yes. And we also compare the expression level of IL-4RA in the humanized mice compared with the wild-type mice, and the results are also yes. Then we try to find out if the mouse can work well, and we use IL-4 to stimulate the mice, and it can secrete IgE. So we found that the action was well, and then we do the efficacy evaluation, and the model can be successfully established, and the positive control can work well and shows a dose-dependent manner. So that's why this model can precisely be a good model replacement for the human disease models. Yeah, thank you.
Awesome. Thanks, Yujie. All right, I think that's all the time we have for questions today. For those of you whose questions we didn't get to, we will be addressing them on our LinkedIn page. So please keep an eye on that. All right, so before we wrap up, I'd like to give a big thank you to Dr. Chen, Dr. Liu, and Dr. Xiong for sharing their insights, and thank you to everyone in the audience for your participation. If you have any follow-up questions or would like to learn more about our humanized models or about AD or asthma, feel free to reach out through our website at biocytogen.com or connect with us on LinkedIn. We would be happy to follow up and discuss your specific projects further. And lastly, just a quick reminder, if you missed any part of today's session, this webinar will be available on demand. You will be able to request a recording on our website very shortly. All right, I think that's all from us today. Thanks again, everyone, for joining, and we look forward to seeing you at our next webinar. Thank you, guys.
Thank you.
Thank you.